today we are I'm super excited because we have a newly minted PhD scientist who is a postdoc and is talking about phytoplankton and zooplankton ecology. So I'm just thrilled to introduce Dr. Tricia Thibodeau. And as we'll see today, Tricia's research spans the worlds of both zooplankton and phytoplankton ecology and physiology. Tricia received her BA from another NESCAC school called Bowdoin College, and she had a major in Earth and Oceanographic Sciences. I went to Hamilton.
Oh, okay. I was going to say, you pronounce Bowdoin very well. Some people don't do it.
Yep. And so Tricia is unique in that she actually was a certified public high school teacher in Maine for a short period of time before she went and pursued her PhD at VIMS with Debbie Steinberg, who some of you guys out there will know. And she studied pteropod ecology and physiology along the Western Antarctic Peninsula, which is one of my favorite locations on Earth. She must be a glutton for punishment because she spent 138 days at sea as part of the Palmer LTER program between 2014 and 2019, and all of it was on the Gould. So she gets points for difficulty in that one. So at VIMS, Tricia received a number of awards, as far as I can tell, as well as including being recognized as the most outstanding PhD student in her class in academics, research, and leadership. She was the co-founder of the Society for Women in Marine Science chapter at VIMS, which is great. We don't have one of those here. Tricia received her PhD from VIMS in 2019, and she's currently a postdoctoral fellow at URI, GSO, as part of Rhode Island's EPS score grant for coastal ecology assessment, innovation, and modeling, where she is working on phytoplankton ecology now. So Tricia has 9 peer-reviewed publications, 6 of which she is first author on, which is outstanding. And it seems from her CV that she's involved in all types of outreach and mentoring as well. So I don't know where you find time for all of that, but it's fantastic. And in her spare time, which you must have lots of, you enjoy biking, cross stitch, and playing with dogs, which scores points for many of us here in the seminar. So with that, why don't you take it away, Trish?
Great. Thank you so much, Dr. Chevenel. And I'll go ahead and share my screen. All right. And should I minimize my little black box here? Can you see that still?
So we are seeing, if you go to your slideshow, that should give us a whole screen. Yeah, there you go.
Okay.
Perfect.
Great.
You're on for it.
Great. Well, thank you again all so much for having me here today. I'm excited to talk with you about environmental controls on plankton physiology and ecology from the Southern Ocean up to Narragansett Bay. And I'll be talking about plankton in 2 forms. And when I'm talking about plankton, I mean passive ocean drifters. And there's 2 main groups. We look at zooplankton, which are passive animal drifters, and then phytoplankton, which are the plants of the sea. And so I'll first be talking with you all today about zooplankton dynamics along the Western Antarctic Peninsula, which we've always referred to as the WAP, but now given Cardi B's recent song on the WAP, I don't know if we can use that acronym anymore, so out for debate. But for the purpose of my talk today, it'll be the WAP still. And I'll be focusing specifically on pteropod physiology and ecology. And pteropods are these really beautiful open ocean snails. A few pictures of them shown here, and I'll give you more background information on them in a little bit. And then we'll be talking about phytoplankton bloom phonology, or the timing of blooms in Narragansett Bay. And these are some of the phytoplankton we see in Narragansett Bay, including a dinoflagellate called Seratium. It has these beautiful horns shown here. and then a diatom called Cattosaurus, which has these cool spines and it's this beautiful green color. And then the most common diatom we see in Narragansett Bay, which is this one here called Skeletonema. And I just need to turn on my pointer. All right. So we're first going to be down in Antarctica looking at the WAP. And it's great to see so many people here at USF that are studying Antarctica. It makes me feel like I'm part of a family here. And so, as some of you may know, one of the reasons we're particularly interested in the WAP is because it's one of the most rapidly warming regions on our planet. This figure here shows winter temperature in degrees Celsius over time, and we see that there's been a long-term increase in winter air temperature at a rate of 1.1 centigrade per decade, which is an overall increase of 7 degrees centigrade since the 1950s, and that's 5 times that of the global average. This region has also seen significant declines in sea ice. This is sea ice duration here in days over time, and we see that there is a lot of interannual variability, including a recent uptick in sea ice duration in the last 10 years. But overall, there has been a long-term decline in sea ice, which is due to later sea ice advance, as well as also earlier sea ice retreat. And since 1979, we've seen an overall 3-month decrease in sea ice season, and this has a profound impact on the ecosystem along the WAP. So the Palmer Antarctica Long-Term Ecological Research Program, or PAL-LTER, aims to understand how these long-term changes in sea surface temperature and sea ice are affecting the ecosystem, all the way from the bacteria and the plankton up to the penguins and whales. And so this is an image of the Lawrence M. Gould. Some of you know here, beautiful bright orange ship. And we take that every year in January, which is summertime in Antarctica, down to the peninsula. And we have been doing this every year since 1993, so almost 30 years now. And we begin our transect at Palmer Station and then move along these series of grid stations shown here in white in a zigzag form, covering an area that's about the size of the state of Oklahoma in 30 days. And what we're particularly interested in getting at is this climate gradient that's developed along the peninsula, in which in the northern part of the peninsula near Palmer Station, it's now essentially a subpolar climate that's warmer and moister than it used to be. And the south region is considered our transition zone. And then the far south region we only added in the last 10 years as sea ice began to open up. And this region is considered our truly polar, cold, dry climate. We also sample across this coast, shelf, and slope gradient, where the coastal region is our most productive and shallow region versus our slope region, that's our deepest waters up to 2,000 meters and is less productive. We also have a sediment trap that is moored in the north shelf of the peninsula. And it's important because it provides our only year-round sampling for our program, in that we deploy it off the ship every January, and then it stays bottom moored at 170 meters year-round, and then we collect it during the following LTER cruise, remove all the cups, put new ones on, and we have a really great seasonal time series. So I'll be talking about that more in a bit. I'll be focusing today on macrozooplankton, which include the krill, salps, and pteropods. We also have a smaller net shown here, which is our 1 meter net that focuses on the mesozooplankton, such as the copepods and the ostracods. But in terms of our macrozooplankton collection, we use a large 2 meter by 2 meter net with a 700 micron mesh, and we deploy it off the back of the Lawrence M. Gould for about 30 minutes at each station. And then we collect all the plankton in this cylinder-shaped cone called a cod end. And then we dump everything in a bucket and count it all on the ship. And we've been doing that since 1993. So we have a really wonderful time series now of zooplankton. And Some of you may be familiar with this, but oftentimes when we're trying to elicit certain zooplankton to be caught in our nets, we have to do dances to appease the zooplankton gods. So this is me doing a pteropod dance in hopes of collecting pteropods for my experiments. And I'm with one of our marine techs here, Josh, who was very happy to do the dances with me. In terms of the types of macrozooplankton we see along the WAP, I mentioned these guys briefly, but we see salps, which are gelatinous zooplankton. They're basically barrel fills of jelly. These little yellow dots you see here are their guts. And then we have the Antarctic krill, which is considered the primary food source for many of the penguins, seals, and whales down in the peninsula and elsewhere in the Southern Ocean. It's about the size of your pinky finger, which is pretty big for zooplankton. But then we have the pteropods, which are actually the third most abundant along the WAP. And there's two main kinds of pteropods. There are the shelled pteropods, which I'll refer to as thigozomes. And these include the species Limacina helicina antarctica, as well as Cleopyramidata. And then we have the non-shelled pteropods, which we call the gymnosomes, and those include Cleone Limacina antarctica and Spongiobranchia australis. And there's actually a really interesting predator-prey dynamic between these two groups of pteropods in that the gymnosomes almost exclusively feed on the thicosome pteropods. And I'll talk about that more in a little bit. Most of my research today is going to be focusing on the species Limacina helicina antarctica. So I wanted to give you a little more background on them. They have a shell that's composed of calcium carbonate in the form of aragonite, which is considered the more soluble form as opposed to calcite. And it's an important grazer of phytoplankton, especially along the WAP, where it has grazing rates that surpass that of the Antarctic krill. And it's also a prey for higher trophic level organisms, especially fish, including the silverfish, Pleuregramma antarctica, in the Southern Ocean. And it's generally considered an omnivorous feeder, and it feeds with a mucus web that it extends and retracts with ciliary action. And this is one of the few photos we have of a pteropod feeding in the open ocean. This is a different species called Coralla. But you can see that its mucus web is extending from its mouth, and then it has particles that are getting trapped on its mucus. Some of you may have heard of pteropods before because they've been heralded as bioindicators of ocean acidification. These are photos of Limacina shells that have been exposed to increasingly lower levels of pH waters, and you can see that their shells are getting increasingly dissolved with that lower pH. And we're particularly concerned with pteropods in the Southern Ocean because it's expected that by 2100, the Southern Ocean may be completely undersaturated with respect to aragonite. This figure here shows that in the pre-industrial era, the oceans were completely oversaturated with aragonite, and we're starting to see some undersaturation in areas throughout the oceans currently. But then by 2100, we'll see almost complete undersaturation of aragonite in the Southern Ocean. And again, aragonite is that building block of pteropods shells. But what I realized when I was beginning my research with pteropods was that there'd been quite a bit done with pteropods in relation to ocean acidification, but we still didn't really have a very good understanding of their ecology and their life histories and their long-term abundances. And so this led me to my main research goal, which was to determine the environmental controls on pteropod ecology and physiology along the Western Antarctic Peninsula. And so I had three main questions. The first was, are there long-term changes in pteropod abundance and distribution in relation to environmental controls? And this research was published in Limnology and Oceanography in 2019. And then I wanted to know how these shifts in environmental conditions may be affecting Limacina metabolism along the WAP. And this work was recently published in the Journal of Experimental Biology, Marine Biology and Ecology. And finally, I wanted to know if there have been shifts in Limacina phonology or life history patterns and what environmental conditions are controlling these patterns. And this work is currently in press in Deep Sea Research 1. So to begin, I wanted to look at this first question of identifying long-term changes in pteropod abundance. And I utilized the time series data we had on pteropods from 1993 and converted these abundances into abundance anomalies. And so to orient you to these plots, we have year on the x-axis and then the abundance anomaly on the y-axis. And any bar that's below the 0 line here indicates below average abundance for that year relative to the overall abundance mean for that time series. Any bar above the 0 line here indicates above average abundance for that year relative to the overall abundance mean for the time series. So if we look at Limacina abundance here, we see that it oscillates over time, but there's no long-term directional trend. So we see that there are these subdecadal oscillations in its abundance. Versus if we look at the gymnosomes, we see that there is a long-term increase in their abundance corresponding to these long-term warming patterns we've been seeing in the WAP. Cleopyramidata, which is another shelled pteropod, has a similar trend to that of Limacina in that its abundances have oscillated over time with no long-term directional trend. I mentioned earlier that gymnosomes are almost exclusively predators to shelled pteropods, and these are some photos that I took from the last cruise I went on in 2019 of Cleone eating one of the Limacinas. And I have videos of this too, but with the bandwidth, I wasn't sure if they'd come through. But what you can see is that the radula, these like literal claws from Cleone, are up in the shell of the Limacina, and it's literally sucking out the life of this Limacina. And this black tissue that you see here is the tissue from the Limacina that's entering the gut of the Cleone. And about an hour and a half after witnessing this carnivory in action, all that was left was an empty but rather pristine Limacina shell and a very happy and full Cleone. And I wanted to see if we could observe these predator-prey interactions in the long-term abundance data we had. And so I was excited to see that we could, and that increases in Limacina abundances corresponded to increases in gymnosomes for that same year. I also wanted to see if what environmental controls were leading to these shifts in pteropod abundance. And one of the main drivers was sea ice. This figure here shows sea ice advance on the x-axis in relation to Limacina and gymnosome abundance on the y-axis. And we saw that there was a positive relationship with sea ice advance and gymnosome abundance, in that later sea ice advance typically led to higher gymnosome abundance. We also saw that for sea ice retreat here, that positive sea ice retreat or later retreat led to decreases in Cleopyramidata abundance. So overall, we saw that lower sea ice conditions tended to favor pteropod abundance. We also saw that the multivariate ENSO index, or the El Nino Southern Oscillation, had an important control on Limacina abundance. This figure here has the MEI index on the x-axis. And what we see is that as the MEI index became more positive or favored an El Nino year, Limacina abundance decreased. And typically we see in the WAP that a La Nina year corresponds to lower sea ice conditions. So essentially what we see here is that lower sea ice is corresponding to higher Limacina abundance. We also saw that more positive sea surface temperature anomalies corresponded to more positive Limacina abundance anomalies. And so overall, we saw that pteropod abundances were best supported by ice-free, warmer waters. We do also have an extensive time series of carbonate chemistry variables collected as part of the PAL LTER. And so I wanted to see if there were any connections with carbonate chemistry and pteropod abundance. And to orient you to this figure, we have aragonite saturation anomaly. So it's important to note that typically when we talk about aragonite saturation, we are referring to an index where anything above 1 is oversaturated and anything below 1 is undersaturated. And this is an anomaly, and so it's important to note that these numbers don't actually correspond to that threshold number. But what we see is that there was no significant relationship with either shelled pteropods in relation to aragonite saturation. And one of the main reasons for this is that we actually don't see any extensive periods of undersaturation along the WAP currently or in the past 20 years. And so we just might not be experiencing these projected conditions yet that are expected to be seen along the WAP. Another thing is that our time series is only 25 years, which, according to some chemical oceanographers, we feel that may not be a long enough time series to really elucidate these cycles in carbonate chemistry, especially since we don't have any seasonal comparison for what's going on. But this is important baseline data for us to understand the dynamics with pteropod abundances over the long term. Overall, we saw that there was interannual variability in abundance, but no long-term decline in pteropods. Most of them were remaining stable or even increasing for the gymnosomes over the past 25 years. And there was no significant influence of carbonate chemistry parameters, such as aragonite saturation state, on abundance. And La Nina, warming, and associated sea ice retreat best explain abundance trends, and this emphasizes that subdecadal oscillations are an important control on pteropod abundance. And I think overall these points emphasize that right now pteropods are doing okay. I think we tend to have this dialogue of a doom and gloom thing for pteropods, but right now I was glad to see that they're hanging in there. And overall, these results increase our understanding of pteropod responses to environmental variability, which is important for predicting future effects of climate change. I then wanted to ask how these shifts in environmental conditions may be affecting Limacina metabolism along the WAP. And one of the reasons we're interested in this is because over the last 30, 40 years, we've seen this gradient set up where the northern part of the WAP has seen a long-term decline in chlorophyll A, whereas in the southern part of the WAP, we've seen a long-term increase in chlorophyll A. And the reasons for this are because in the northern part of the WAP, there's no more year-round or perennial sea ice. And coupled with increasing temperatures, the winds are now able to sustain over that open water for longer, which is increasing the relative mixing that phytoplankton are experiencing. And so they're getting mixed below their critical depths and therefore aren't able to bloom. And so we're overall seeing a long-term decline in phytoplankton biomass or food for zooplankton. Versus in the southern part of the peninsula, where we still have year-round sea ice, coupled with increasing temperatures and periodic releases from light limitation through this periodic loss of sea ice, we're seeing an increase in chlorophyll, which is represented as an increase in food for zooplankton. And so I wanted to see how these differences in temperature and food might affect pteropod metabolism. And so to do this, I set up multifactorial experiments where I did a number of zooplankton pteropod dances on the back deck to collect pteropods for these experiments. And then at the same stations where I collected these pteropods, I did a CTD cast, or a conductivity temperature depth cast, with Niskin bottles, where I collected high chlorophyll for my high chlorophyll, high food treatment at the chlorophyll max. And then I diluted that water down to a 10% concentration as my low food concentration. I then acclimated the pteropods at these 2 food treatments, as well as at 2 temperature treatments. My low temperature treatment, which was considered my ambient temperature condition for the summer surface waters in the WAP, and then my anticipated future temperature condition of 4 degrees centigrade. I then incubated the pteropods for 12 hours and took metabolism measurements during that time, which I'll talk about in a moment. Also did one CO2 experiment to look at how ocean acidification may affect pteropod metabolism within this same 12-hour time span as temperature and food. And so to do this, I had a CO2 and a zero air cylinders that I mixed with mass flow controllers to create a high and low CO2 experiment or treatment. And then I collected water again from the CTD, and I filtered them to remove any phytoplankton and bacteria, and then I bubbled them to create these high and low CO2 conditions. And then I incubated the pteropods for 12 hours in these 2 conditions. And I just wanted to give you a sense of, oh, I'm sorry, one more slide. In terms of the measurements I was taking for these experiments, I would measure their respiration or their oxygen uptake every hour for 12 hours, and I was using a Pyroscience fire sting system shown here. And I had a fiber optic cable that I would connect to an optical dot that I glued onto each bottle. And that would allow me to shine a light basically and measure the concentration of oxygen in the bottle for each hour. And then at the end of the experiments, I would filter out the water for chlorophyll, thiopigments, fecal pellets, and then collect the excrement or their pee so that I could measure what they had excreted in those 12 hours. So that included measuring things like dissolved organic nitrogen and carbon, as well as ammonium and phosphate. Overall, I did 7 experiments throughout 3 cruises from 2017 to 2019, and I tried to capture this north to south climate gradient that we see along the peninsula, as well as this coast to slope gradient we see from on to offshore. I had an overall temperature range of negative 0.3 to 5.5 and a food range of 0 to 6.28 micrograms per liter. Just to show you a few results, we saw overall that there was an increased metabolic response under higher temperature conditions, but not really under higher food conditions. This figure here has temperature on the x-axis and then the corresponding food concentration on the y-axis, and the respiration rate here on the other y-axis. And what we see is that as temperature increases, the respiration rate increases as well, but there was no significant effect of food on respiration rate. We also saw that excretion rate significantly increased with increasing temperature with minimal effect of food. This is ammonium excretion here on the y-axis, and we saw similar results for that of phosphate as well, but we did see a significant effect for food on dissolved organic carbon, dissolved organic phosphorus, and urea excretion. We also looked at physiologic metrics to determine metabolic responses of pteropods to shifts in temperature and food. And so this is the metabolic oxygen to nitrogen ratio, which is looking at oxygen respired to nitrogen excreted. And we see that with increasing temperature, the O to N ratio decreases with no significant effect of food. And what this means is that under increasing temperature conditions, pteropods were favoring usage of their short-term energy reserves, which includes that of proteins, as opposed to their long-term reserves such as lipids and carbohydrates. We also saw an increased proportion of total nitrogen excreted as dissolved organic nitrogen with increasing temperature. This has temperature here on the x-axis and then percent nitrogen excreted on the y-axis. And we see that especially under the higher temperature conditions that more of the nitrogen is excreted as dissolved organic nitrogen as opposed to inorganic nitrogen in the form of ammonia. We did not see any significant effect of CO2 on pteropod respiration or excretion. This figure here has respiration rate and excretion rate in ammonia on the y-axis, and then the high CO2 treatment and the low CO2 treatment. And we saw no significant difference between the two, which was somewhat to be expected given what's been done on pteropod metabolism under CO2 in the past, in that typically it seems to take at least 24 to 48 hours to elicit a significant effect of CO2. But I thought it was still important to conduct this experiment so that we could understand the full range of environmental conditions that could affect pteropods within this 12-hour short-term time frame. So overall, we saw that the highest metabolic rate occurred under higher temperature conditions with less effect from food and no effect from dissolved CO2. The metabolic O to N ratio decreased with increasing temperature, with an increased reliance on short-term energy reserves. And there was an increased proportion of organic nitrogen, as well as organic phosphorus, which I didn't have time to show under higher temperature. And we also saw that the C to N to P excretion ratios were all below the Redfield ratio. And this has important implications when we think about dissolved C to N pools and the food resources for the microbial communities in the WAP. And this work increases our understanding of the role pteropods play in biogeochemical cycling in a warming ocean, particularly since we don't have many measurements at all, especially of excretion for pteropods in the Southern Ocean. Moving on, the the last question I had was looking at if Limacina phenology patterns have shifted over time and what environmental conditions control these patterns. I wanted to orient you more to this concept of phenology. It's the study of annually occurring life history events. And I thought this animation by the New York Times was really good, and I hope it comes through on your end. But essentially what we're going to look at is the spring foliage of 2017 when it first begins. Let's see if it'll, oh, I guess. And so we first see that the spring foliage begins in January in the south and extends to begin all the way up in the north in April and May. And what we can do is compare timings in foliage for a year relative to the overall historic timing of that foliage. And so what this figure shows is the comparison of that timing in spring foliage in 2017 to the 30-year average. And what we see is that in the southern United States, including in Florida, the spring foliage is occurring up to 10 days later than it used to 30 years ago, versus in the West Coast and in the Northwest, or in the Midwest, we see that the spring foliage is starting up to 10 days earlier than it used to. And I wanted to apply this same concept to pteropod phenology. So to do that, I utilized a sediment trap, and typically sediment traps are used to collect sinking particles, mainly detritus, zooplankton carcasses, fecal pellets from the surface to the deep oceans so that we can better understand carbon export. But pteropods will often fall into the trap as well because they are negatively buoyant. Those that have shells, oftentimes as a defense mechanism, they'll retract into their shells when they come in contact with a predator. And so we think what's happening is they are floating along and then they hit the sediment trap and they go, oh no, and then they retract into their shells and they sink and they get caught forever. But it's great because they have this really nice seasonal time series for us to look at over the long term. And so our sediment trap is located, like I said, on the northeast shelf here. And it's bottom moored, so it rests 170 meters below the surface. And it's this giant yellow cone that you can see here. It's about my height. And we deploy a series of cups that rotate around the sediment trap at intervals of about weekly to biweekly basis. And they collect any of the sinking detritus and pteropod shells that are falling into the trap. And so I used the sediment trap time series from 2004 to 2018 to analyze the pteropods shells that were in the trap. And so to do this, I picked all of the pteropods from the cups and measured their diameters here to look at their lengths. And I had a ton of help on this from a William & Mary undergrad, Colleen McBride, who is a well-deserved co-author in this paper. I figured out that she counted and measured over 10,000 pteropod shells during the course of 2 years helping me. So I am forever indebted to her. Thank you, Colleen. But what we got from this data set was really exciting. This is a size frequency histogram, which is showing the different lengths, the abundances of different lengths that we saw for each year. And so this is just an example from 2017 to 2018. And what we saw was that typically we first see a new cohort of pteropods appear in the sediment trap in June, which is about early winter. And then they're continuously growing throughout the winter season. And this was really exciting to see because we didn't know yet if pteropods actually grew during the winter months. No one had measured that before. And then they stopped showing up in the sediment trap around January. We do occasionally get some larger adult pteropods that fall into the sediment trap, but often we don't see them. And we think that's because they're feeding in the surface waters during that time. And then what we think is happening is that around April or May, after they've been spawned, the juvenile pteropods are sinking into the deeper waters as a form of ontogenetic migration, which other zooplankton like krill do. And then they're ending up in the sediment trap around June and growing again throughout the winter season. And so what I was then able to do was take the median shell lengths from each of these different cups, which is what these different lines here represent. And I was able to determine growth rates for the pteropods for each year of the time series. So we have the growth rate here on the y-axis, and then the month here, and these are the different cups, the median length for each cup over time. And then I was able to compare those growth rates over time to see if there had been any change in their growth over this time series. And what we saw was a lot of interannual variability in that some years there was really fast growth, and other years, just a couple years later, there was slower growth. And I also compared the growth rates looking at juveniles versus adult size classes. But overall, we saw that there was no long-term directional trend in pteropod growth over the time series. I also looked at the time of appearance that pteropods first began falling into the sediment trap every year. And again, we saw there was a lot of interannual variability. Some years, like in 2006, the pteropods showed up really late as July. And then other years, they were showing up really early, like in 2012, as early as February. And overall, again, we did not see any long-term directional trend in their time of appearance, which indicates to me that based on their growth and time of appearance, that their phonology hasn't significantly changed over this 14-year time series, although there is a ton of interannual variability. And so I wanted to see what environmental factors might contribute to this interannual variability. And again, we see that sea ice and sea surface temperature are important controls. So this figure here shows sea ice retreat on the x-axis and time of appearance on the y-axis. And we see that later sea ice retreat corresponds to later pteropod time of appearance in the sediment trap. We also see that more open water corresponds to a higher growth rate or growth coefficient. And we see that sea surface temperature is also important in that this figure here shows autumn sea surface temperature on the x-axis and then growth coefficient on the y-axis. And we see that it's a positive relationship. And the same with winter sea surface temperature in that a more positive sea surface temperature corresponds to a later time of appearance. And so overall, we see that earlier time of appearance is related to earlier sea ice retreat and higher sea surface temperature and lower primary productivity, which I didn't have time to show. But this means that generally we're seeing earlier retreat corresponding to earlier migration into deeper waters and therefore earlier appearance in the sediment trap. We're also seeing that growth is related to more open water and higher sea surface temperature, as well as chlorophyll A, which again, I didn't have time to show. But this means that overall, we're seeing warmer, more open waters are promoting faster pteropod growth. And so again, we're seeing that sea ice and sea surface temperature are playing an important role in pteropod ecology along the WAP. So to conclude, we saw that this is the first study in Antarctica to show pteropods actively grow during the ice-covered winter season. And less sea ice, higher sea surface temperature, and food are important controls on pteropod phenology. And changes in time of appearance and growth have important implications for the regional food web when we start to think about match, mismatch, and certain types of species like fish, for example, that might be timing their spawning events and whatnot on pteropod abundances for food. And these resulting changes also affect the organic and inorganic carbon flux from the surface to the deep ocean, given that pteropods are made of calcium carbonate, and so therefore their shells and their shell dissolution can affect the solubility pump in the WAP. And so overall, I argue that pteropods are not only bioindicators of ocean acidification, they're bioindicators of climate change in general, because we see that they're not only responsive to ocean acidification, but they also respond to sea ice, as well as temperature changes and shifts in food on subdecadal scales. And this also emphasizes the importance of these time series and being able to elucidate these long-term trends and the importance of these subdecadal oscillations on pteropod and zooplankton ecology in general. And this is why I think pteropods are so terrific. With that, I would like to acknowledge the Coastal Ecology Assessment, Innovation, and Modeling Program. It's a Rhode Island EPSCoR grant that's funding my postdoc, as well as the Rynierson Phytoplankton Ecology Lab at URI that I'm a part of. I started my postdoc in January, right before COVID happened, but this is about as good as I can get for a lab photo right now, as I'm sure many of you can relate to. I'd also like to thank my funding sources during my PhD, the NSF Office of Polar Programs, as well as the Virginia Institute of Marine Science, and the Steinberg Zooplankton Ecology Lab, run by the wonderful Ms. Debbie Steinberg, or Dr. Debbie Steinberg. And I just want to thank you all for having me here today. I really appreciate this opportunity. And with that, I will take any questions. Thank you.
Thank you, Tricia. That was awesome. Really great talk. And I am really impressed by the murals and the work that you did at RISD as well. That's really, really cool. So I want to open it up for questions. But right now, what we'd like to do is we'd like to have students ask the first couple of questions. So any students out there have any questions for Tricia? Anyone? Raise your hand. Just start talking. Brianna, I saw your hand first, although Alex. No.
There we go. Press mute twice. Okay. Hi. I was just wondering. You said, if I understood correctly, that the pteropods' growth rate seemed to increase with higher temperatures and less sea ice cover. But you were also saying earlier that the lower sea ice cover was associated with less phytoplankton. So I was curious what was going on there. Is it like the location of the sediment trap, or yeah? So I was kind of curious because it almost seems like counterintuitive.
Yeah, no, I think it's a good point. I mean, it's important to note I was sort of jumping different time scales here. So when I'm doing my experiments, those are on really short time scales of just 12 hours versus when I'm working with the sediment trap, I'm thinking at interannual time scales. And where the sediment trap is located, it is seasonally covered by sea ice in the winter time. So it does experience that sea ice coverage. But I guess in terms of their phenology, what I mean is that it does seem like that phenology of when they enter the sediment trap is tied with the timing of that retreat, and that if the sea ice is retreating earlier in the year, that means that they're showing up in the sediment trap earlier as well.
Thank you.
Yeah.
Okay, Alex.
All right. Thank you for your talk. I really enjoyed it. I have a few questions, if you don't mind, about your metabolic experiment, and then the second one you talked about. So I'm in Dr. Seibel's environmental physiology lab, and we do similar things with plankton and cephalopods and whatever. So I'm really interested in this kind of topic.
For sure.
And so my first question is, you said that you took optical oxygen measurements every hour. So did you was your metabolic bin size an hour, or did you just take one reading every hour?
I just took one reading every hour because I had 24 bottles total. And so I wasn't able to continuously measure them, which would have maybe been the ideal. But I did also do a couple test runs where I did measure continuously just to make sure that I was catching that decline appropriately. But yeah, I was just measuring them every hour and taking a few measurements over a minute.
Oh, very cool. Thank you very much. And my second question is, you said that metabolic rate was not affected by food concentration. So did you incubate the animals with food in the chamber with them, or did you take them out of the food treatment and then put them in the chamber?
I did. So I acclimated them with food, and then I incubated them with food as well. And that was one of the reasons I kept the experiment to 12 hours, because I was a little concerned about running it longer and having issues with the phytoplankton growing or something like that and messing up my results. But I did have controls as well, of course, with phytoplankton in them, that I was able to also get a sense of the difference in oxygen concentration between the two bottles.
All right, and then my final question is, are you assuming that the animals were at, I'm assuming that you're assuming that the animals were at standard metabolic rate when you were taking this oxygen measurement? Is that true, or is that not? Yes,
I think that's true. And I did also do some experiments I didn't mention where I did just measure their standard metabolic rate without doing any manipulations, so that I could also compare my manipulations to those more standard experiments.
Okay, and I guess my follow-up is, what was the lowest PO2 that you got during the trials?
Oh gosh.
I
I can look that up and email you after.
Oh, okay,
if
cool.
that's
We'll
okay.
talk later. Thank you.
Yeah, sure. Thanks for all your questions.
Okay, Brianna, did you have a second question, or is your hand just still up?
I didn't mean to have my hand up, but I actually do. I was wondering what the error bars on the decadal time, when you averaged by decade. I was curious how much separation there was in terms of the error bars on those decades.
Yeah. So when I compare the first and the last decade in terms of the start date, there is a significant difference of about 1 month, which I said. But then when I didn't actually see a significant difference in the timing of the max bloom over the different decades. And when I compared the start date between, for example, the 1980s, I think, to like the 1990s, I didn't see a significant difference. So they weren't all significantly different, but the first and last decade were, which I thought was maybe the most important.
Cool, thanks.
Mm-hmm.
Okay, before we move on to more general questions, are there any more student questions? Okay, anybody else have questions? Raise your hand, jump in. Brad.
Oh, yeah, Brad.
Let's see, unmute. There we go. Hi, Tricia. Great to see you.
Hi, Brad. Great to see you.
Super fun.
I didn't know if I should give you a shout out in my talk or not.
No worries. But great to see where you've taken all this. That's great. I just had a couple quick questions. I was wondering, you mentioned that the pteropods ontogenetically migrate. I'm wondering if the carbonate saturation changes much there with depth, and if that could influence any of your interpretations.
Yeah, I don't have any carbonate chemistry data at the sediment trap location, so it's hard for me to say exactly if they would be affected. But a paper by Claudine Hari in 2012 summarized a lot of the carbonate chemistry data from the WAP, and she did see undersaturation of aragonite in wintertime in some of the deeper locations. So potentially it could have an important implication on the early life histories of pteropods for sure. But I unfortunately wasn't able to test that with my data set.
All right. My second question, maybe I just missed this, but were you able to verify that the animals in the high food concentrations actually ate, or were they just incubated and was that?
By making sure they ate, do you mean like checking their guts or
concentrations of phytoplankton before and after or something?
Yeah.
I don't know.
Yeah. I did measure concentrations of phytoplankton before and after, and yeah, to confirm that they were eating, and they were sometimes producing, pseudofeces and things like that. So at least that made me feel like they were digesting something. And I did also try to make sure that I was starving them for a little bit before the experiment to empty their guts ahead of time before I did the experiment.
Could you see if they were actually putting out a mucus web at all?
It looked like they were doing some like mucus creation, but no extensive webs, which at least because Amy Moz was a collaborator on this project with me, and she didn't seem to think that they would do any mucus web production in captivity, and I don't think I saw it in the time of my experiments, unfortunately.
I know Amy was successful without a Woods Hole with local animals around there, so but we never were at McMurdo. I think we had them in flowing seawater and they would just get all tangled up and it was a mess. So
yeah.
All right, thanks. Well, good to see you.
Yeah, thank you. You too.
Okay, anybody else have a question? I have a question while we're waiting for other people. Well, I also might have an answer to Brad. So what depth is your sediment trap at? I used to know this information and I don't anymore.
That's okay. It's 170 meters.
Okay, so you should be right at, depending on the time of year, the interface between CDW and winter water, in which case you would have a change in carbonate ion concentration, I think.
Mm-hmm.
So it would be lower.
Yeah. I mean, I very well think that could be the case. I think, well, so I was going to say, unfortunately, we don't have the data for it, but Elizabeth Shadwick actually recently put out a mooring with a PCO2 sensor on it right near the sediment trap location. And she's currently working on analyzing the data now. So that'll definitely give us a much better sense of the dynamics going on there.
Cool. I don't know if you know, but I look at the sediments right below the sediment trap all the way to the last deglaciation.
Well, I met one of your students at the lunch, and I wasn't sure with Antarctic margin where exactly on the margin that was, but very cool. I bet you get some old pteropods in there then.
We don't. They dissolve before we, like, if we do see them, they're dissolving as we're sampling. So it's a bummer. But if you have any extra pteropods, we're happy to analyze them for you.
Oh, cool. Awesome.
So, yes. Anyone else have any questions? No?
I did see one question. Oh, yeah.
Tracy, go for it.
All right. Oh. Am I on? Okay. Hi, Tricia. I don't know if you
Hi.
that I was there at Palmer in 2015.
Yeah, the first year I went, I remember.
Yeah. So, yeah, I have like a kind of boring method question for you, but I'm also working on an Antarctic paper from the 1990s data and using the ice retreat. And I'm just wondering how you decided when ice retreat starts, because sometimes it's like up and down, and it's hard to, like, how do you decide among all that crazy ice data what counts as retreat?
Yeah, it's based on Sharon Stammerjohn's 2008 paper, where she talks about sea ice dynamics, and she essentially defines it as, I believe it's like March 15th is the start, and then like March 1st of the following year is the end, and that's just based on, I think it's like when sea ice concentration like retreats below 15% of the average. I'd have to double check the exact numbers, but I'm happy to send you the paper that discusses that.
All right. And I will look for it, and if I can't find you, I will find it. I'll let you know.
Yeah, for sure.
Thanks.
Yeah.
Anyone else? Oh, Frank.
Hi, Tricia. How are you doing?
Hello. Good. Thanks.
Yeah, good. Good talk. I was curious about your phenology work in the phytoplankton concentration in Narragansett, and I don't, maybe I missed it, but I don't know if you talked about what you attribute any of the changes, but it probably is so complicated by nutrients, the human aspect, and any changes in wind. So and I was also curious exactly how you chose what you decided to be the first point that you considered an increase, because and it went by so quickly, I kind of missed it.
Yeah. So in terms of the metric I use to identify bloom start date, it's based on a phenology metric that was developed by David Siegel in 2002, where he identified the bloom start date as crossing being 5% above the median chlorophyll value for that year. And I have played around with some other bloom phenology metrics, like looking at the cumulative sum of chlorophyll over time and things like that. And it seemed like the Siegel metric was the one that was most commonly used. So that's what I've been working with. But your point about variability, it's definitely taken me a while to get my hands around Narragansett Bay because it is so variable, and the chlorophyll even is just like all over the place. So I've been working with a statistician actually to develop these dynamic regression models that'll help us better constrain the blooms every year. And that's actually the data that I used, that I showed for the phenologies. But you're right, I'm still trying to figure out what's going on with the environment. And I think ultimately it'll probably be sort of what you said, where there's just so much variability. It's really hard to parse out significant predictors, particularly for phenology, I think. So yeah, I think it'll be something I'll need to look into more in the future for sure.
Thank you.
Yeah, thank you.
Okay, anyone else? I think if there are no more questions, we should probably thank Tricia for giving a wonderful seminar, and in my favorite place in the world, mostly. And yeah, so thank you very much. We really appreciated you joining us today.
For sure. Thank you for the opportunity. And I enjoyed meeting with the students today at lunch too. That was fun.
Excellent. Good.
Thanks, everybody.
Thank you, and we will talk to you later.
Sounds
Bye.
good. Bye, everyone.
Bye, Tricia.
Great to see you all.